DNA replication is important to say the least. It is because of DNA replication, hereditary genes are able to be passed down from generation to generation. During mitosis and meiosis, both processes to divide cells, DNA replicates in the nucleus of a eukaryotic cell and the cytoplasm of a prokaryotic cell during interphase, more specifically, the S phase. Before I begin to explain the process of DNA replication, you must have some background knowledge about DNA. DNA is made up of nucleotides. A nucleotide consists of a phosphate group, a deoxyribose sugar, and a nitrogenous base, those including adenine, guanine, thymine, or cytosine. Adenine and guanine are purines. This means they have 2 carbon rings, whereas thymine and cytosine are pyrimidines, meaning they only have 1 carbon ring. To make a double-stranded helix DNA, one purine will form hydrogen bonds with one pyrimidine, and according to Chargaff's rule, adenine will always pair with thymine with 2 hydrogen bonds, and guanine will always pair with cytosine with 3 hydrogen bonds. Because adenine and thymine are bonded with less hydrogen bonds, it is easier to break them apart. Further, the number of adenine will always equal the number of thymine, and guanine with cytosine because of this base pairing rule. Now, DNA replication. The 2 strands of DNA are anti-parallel to one another. This means one strand goes from the 5' end to the 3' end, whereas the other strand does the opposite, going from the 3' end to the 5' end. DNA replication cannot take place without the help of enzymes. The process begins as helicase unzips the DNA through breaking the hydrogen bonds connecting the nucleotides. To ensure the remaining DNA, yet to be unzipped, does not supercoil, topoisomerase stays at the replication fork. And in order for the now separated strands to not bind together again, single-strand binding proteins are placed. Now that the 2 strands are separated, primase lays down a primer made of RNA that is anti-parallel to the respective strands and will guide DNA polymerase III on where to begin. DNA polymerase III creates the new complementary strands of DNA moving from the 5' end to the 3' end. One strand, known as the leading strand, will face no issues as DNA polymerase III can work continuously on building DNA as it uncoils. However, the other strand, known as the lagging strand, will work at a slower pace. This is because as the DNA further uncoils, the original primer set will not cover the whole strand, because DNA follows a directionality of going from the 5' end to the 3' end. Therefore, primase will continue to lay down primers as the DNA strands uncoil, creating fragments, further known as Okazaki fragments. Then, when the DNA is all uncoiled, DNA polymerase I will remove each unnecessary primer on the lagging strand to create a continuous 5' to 3' strand of DNA. With these gaps comes ligase to fill the holes left by the primer and thus completes the replication of DNA. The end product of DNA replication are 2 double helix DNA molecules. DNA is replicated in a semi-conservative fashion, meaning each molecule is made up of 1 original strand and 1 new strand. And that concludes DNA replication. Thank you for watching.